Hello students, this is Dhayabaran from the Department of Chemistry, Vishwanagar College. I am happy to present a small learning object on power of protein and its functional properties. Energy is an important element that must be present in every walk of our life. It helps to lead a happy and vibrant life. Yes, whatever our mind conceives, that will be achieved by a physical body. It is important to have energy in every walk of our life. It is interesting to know that protein serves as a versatile, multifunctional unitary source. It builds our body structure, acts as a catalyst and transport ions, and build our immune system and transmit nerve pulses and controls growth. The three-dimensional structure of protein from amino acid sequences play a vital role in structure, functioning and stability of the molecule. The protein molecule not only serving as energy reservoir but also acts as a basis for fundamental basis of structure and functioning of the body. From this small learning object, we can understand the influential role of chemistry in terms of structure of protein, which can bring out lot of information about biological functioning, as well as we can understand how the change in structure of the molecule can bring lot of influences on the biological functioning.
Eat healthy, think better is the slogan of Britannia. Higher order of thinking is the order of the day. A sound mind thinks better. The lifestyle and eating habits will make a big difference in life. It is possible to train our mind and body to build our stamina by making smart choices of food items. Lean meat, egg, fish, milk products and seafood are rich sources of protein. Protein consists of amino acids linked by peptide bonds. Each amino acid consists of key elements carbon, nitrogen and oxygen. In addition, the carbonyl carboxyl group and side chains are attached. Difference in side chains distinguish the various amino groups. Different side chains determine the properties of 20 amino acids. The linear sequence of amino acid within a protein is considered as primary structure. It is built for a set of 20 amino acids. Secondary structure refers to regular recurring array of adjacent amino acid residue in polypeptide chain. It is maintained by hydrogen bond between amide hydrogen and carboxyl oxygen of the peptide backbone. The two major secondary structures are alpha helix and beta structure. Protein needs to adopt three-dimensional shape in order to function properly. Chains of amino acids folds into three-dimensional structure. Some of the proteins are composed of two or more polypeptide chains referred as subunits. The spatial arrangement of these units is known as quaternary structure. Ramachandran plot serves as a tool to probe the structure. Ramachandran plot is also known as Ramachandran diagram, which was originally developed in 1963 by G.N. Ramachandran. The angles, dihedral angles psi and phi are plotted in this diagram. The conformation of peptides are defined by the values of phi and psi. The rotation of the bonds N-C alpha and carbon-carbon bond will be recorded. Each peptide bond has partial double bond character due to resonance and cannot rotate. Three bonds separate sequential C alpha in polypeptide chain. The N-C alpha and C alpha C bonds can rotate with bond angles designated as phi and psi respectively. The peptide CN bond is not free to rotate. Other single bonds in the backbone may also be rotationally hindered depending on the size and change of the side chain. Both phi and psi increases as the carbonyl and amine nitrogen rotates clockwise. Values of phi and psi are plotted in this diagram for two-dimensional structure of protein. A Ramachandran plot can be used in two different ways. Number one, to show the theory which values or conformations of the phi and psi angles are possible for an amino acid residue in a protein. Number two is to show empirical distribution of data points observed in a single structure in usage for structure validation or else in a database of many structures. The applications of Ramachandran plot are: number one, to determine protein structure and the position of amino acids. Number two, if the percentage of an amino acid is more than 88%, it is allowed region. It can be used for in silico studies. Number three, it tells about allowed and disallowed regions of the protein that reflects the stability and structure of protein.
From this small learning object, we can understand, number one, the influential role of chemistry towards structure of protein, which influences the biological functions of protein. For example, if there is any error in the amino acid sequence of protein, that will lead to health issues like malnutrition and genetic disorder diseases. Number two, we can appreciate the contribution of the Indian scientist Dr. Ramachandran. It is quite appealing and informative for his valuable information towards the structure and function of protein. Considering him as an emulative example, the younger generation can be motivated and impressed to go further in explorative studies and contribute more scientific information for the welfare of the human.